start.
I think you have recently launched the health technology assessment module, HTA. So that is good.
Sorry, Vijay, say again.
I said I received an email from your team that you recently added one more features to the nested knowledge.
Oh, yes.
That's most interesting part.
Yes, yeah. I'm going to show you one of the features today, actually. Okay, I think we can start. So before we begin, I'll briefly introduce myself. I'm Jade Thurnham, the Director of User Experience and Design at Nested Knowledge, but I like to come on these calls and help give some demos and show general features of the software. And I think Kevin Kalmus just joined. He's our CEO, sorry, not CTO. I'll let him introduce himself.
Absolutely. No, I do not have the technical capabilities to be our CTO, but I'm the founder and CEO of Nested Knowledge. I'm a researcher myself by background. Previously, I worked as a research consultant to physicians, to hospitals, and to device and pharma companies. And Nested Knowledge was really born out of the struggles we had in trying to execute those reviews using effectively Microsoft Excel. So with that, I'm here for questions. I'm here to demo anything that goes beyond the norm, but Jade, I'll throw it back to you.
Perfect. Thank you, Kevin. And if anyone has any questions at any time, if you could put them into the chat, and then we'll address them as they're appropriate. And Kevin, if you could look at the questions as they come in, and stop me because I won't be able to see since I'll be sharing my screen. That'd be great.
I will pull out any questions that come in the comments and throw them to you when appropriate.
Perfect. Okay. Thank you. Okay, we can get stuck in. I'll share my screen.
So hi, Kevin. Thank you again that you are spending some time with us.
Absolutely. I thought that the webinars and the contacts around this have been really positive, and it's really great to see so many users. We're very focused on collaboration. It's inherently a software that's built around collaborating across teams, and to see such a large team of people who have experience in evidence synthesis, people who have the need for a systematic review software, that just gets us excited. So I've been attending pretty much whenever I can.
Thank you. So our team it's a young team, and most of them are excited to use this nested knowledge for their meta-analysis. And we are today morning I received a mail from either you or your team that you have included the HTA component to the nested knowledge, so which is very interesting.
Absolutely. I'm much more of an academic by my long-term background. I published a lot of systematic reviews before I learned what HTA meant. But believe it or not, the largest commercial use case right now for nested knowledge is HTA, since it's so dependent on systematic review, and since so many people are working so hard to put together these HTAs. And for those who have less experience with HTA, it's effectively how you can argue that a certain therapy is effective and cost-effective for an indication, and it's usually some evidence synthesis combined with sometimes in proprietary data, and it's submitted to insurers. So that's a common use case for nested knowledge on top of academic reviews. But Vijay, do you have something else to add there?
Oh, no, I was just going to say, I'm going to briefly go through the demo nests that we have, and one of them is the one that's dedicated to meeting HTA guidelines. So anyone can access that has an account and can see for themselves.
Vijay, anything else to add there before Jade does jump in?
Yeah. So maybe we can wait for another 1 minute more, because people are still joining. If they miss something at the beginning, they'll be difficult to follow. So we'll start at exactly 9:05.
Perfect.
Sounds good.
Team GCS, if you have any question, you can utilize this 1 minute to ask Jade or Kevin. Shivaji Gupta.
Good evening, sir and madam. Maybe it's too early to ask this question, but I was reading through the first two help documents regarding screening and all. Please just throw some light about how is it different from a regular PubMed search and nested knowledge. We use the same booleans and all in PubMed. So how is it going to help me in the future? Thank you.
Great question. And
Shivaji and all members, just a request, since they do not know us because this is our second meeting. So before asking any question, just introduce yourself, who you are, what you are doing, and which part of the India you are. Because all the members are, because India is so vast, so it will be good. And your domain also, whether you are a medical doctor, public health professional, or any other domain also, that will be great.
Okay, I'm Dr. Shivaji Gupta, Assistant Professor in Midnapur Medical College. I'm pursuing public health, mainly interested in oncology and NCDs.
Thank you, Dr. Gupta.
Thank you, sir.
Great question, Dr. Gupta. And on the search, you will use similar or even the same Boolean operators and Boolean strings to do your search. With nested knowledge, you can set those up as repeatable searches on PubMed. You can also just run it the same way you always would and then bring in the results. So you can either run the search independently and bring it in, or you can run it on nested knowledge. The benefits of running on nested knowledge, we do help you figure out what to put in your Boolean search. We have some tools for exploration of search. And then also it's repeatable. So you can set a repeat cycle where it searches every day and brings in the new evidence automatically. And I think you had a second question about screening and how does it differ from other systems. We will actually be going to that in detail, but the most exciting way it differs is that we do have some AI assistants that can make your screening go a lot more rapidly as soon as you've trained in a robot on how to assist you with screening.
Yes.
Okay, so what I get, it's fine. So what I get is it's going to update itself regularly on its own, so that we don't have to repeat the search in the future. It gets automatically added, right?
Yes, exactly. And I just wanted to say, excuse me, if you couldn't make the webinar that we had a few weeks ago, Kevin actually did a full demonstration of the software. So we can definitely send that link out to everybody to watch after this. But this particular demonstration today is just going to be focusing on the flexibility of the features in nested knowledge versus doing a demo of everything that we offer. So we can definitely send out the link afterwards so you can look at Kevin's demo for each of the features.
Yeah, that will be very helpful. Thank you.
Yeah, of course. Okay. Vijaya, do we have a good number of people that have joined?
Yes, we can start now.
Okay, perfect. Okay, so I'll actually put in the chat the link to our website. So if you want to head to this link, this is, if you haven't already, this is where you will sign up. And once you've created an account, this is where you'll sign in. And so this is what it looks like once you click on that link. To sign up, you just go to the top right corner. And this is if you haven't done it already, but I'll just briefly go through it now. You would hit sign up, fill in your details. On the next page, you'll fill in your email, you'll get a verification email. And once you've done that, you come back to the same page and you hit sign in. I have an account through my Google account, so I just go to continue with Google. And then it brings you to what we call your auto lit page. And this is essentially just a list of all of your nests. And a nest, for reference, is what we call at Nested Knowledge a name for a singular project in the software. And once you've created your account, you're going to want to join the GCES organization. In order to do that, you go to the top right-hand corner, you click on your name, and you go to organizations. And you're going to go to join organization, and you're going to type in the Global Center for Evidence Synthesis. And You're going to want to join a specific one. There are quite a few here. And so actually, if anyone has created any of these organizations, if you could go ahead and delete the organization, because ideally there should only be one. And Vijaya is the admin of this organization. So if you specifically type in GCES-Global Center, you'll be able to find the specific GCES organization. Once you've done that, Vijaya will approve your request, and when you come back to your AutoLit page, you will see the nests that either you have created, so whether that's whether you've already started a nest or not. But if you click on this drop down, you will see the organizations that you're part of. So if you click on GCES, this will give you a list of the nests that are within the organization. So currently there's only one nest in here, but once everybody starts joining the organization and creating their own nests, this is where they'll be listed. So that was a little bit of admin just to start off with. But when you're on this page, you have two options. You can either create a new nest, or you can look at our demo nests. And in terms of the flexibility of nested knowledge, you can look at 3 different types of nests that currently exist in the software. So here we offer a COVID-19 nest that looks at different antivirals. This is a typical systematic literature review and meta-analysis, so you can look at this one. We also have a nest on non-small cell lung cancer, looking at different inhibitors and how they were tested on this type of cancer. And this nest was specifically set up to meet HTA guidelines, or specifically the NICE guidelines, which are the HTA guidelines in Europe. So if you're interested in doing a review that meets those guidelines, you can look at this demo. And then we also have another literature review, which looks at heart failure and the different drugs tested for heart failure. So this is free to anybody. You can open your own demo and play around with it because it will be owned by you. So you can experiment with these different nests. Okay, but for today, I'm going to look at a particular nest that we have, which looks at stroke treatment. Okay. So in any nest, you have these modules at the side, and these work consecutively. So you'll start by doing your search before you do your screening, before you do your tagging or your data extraction, which then generates your outputs, which is synthesis. Again, Kevin goes into more detail in his demo, which we can send afterwards. But for today, I'm just going to show you a few features that, again, demonstrate the flexibility of different project types and what you can do in the software. First, I'm going to start off with search exploration. So if you go to the left-hand side, underneath search, there's search exploration. And this allows you to type in different concepts and see what is out there in the literature. So in this particular nest, these were the terms that were typed in. We typed in stroke. We were interested in the outcomes of mortality, different types of treatment. And once we hit refresh exploration, we get all of these different terms that are generated by what we call RoboPico. And this is a machine learning tool that scans the literature in PubMed and tells you the frequency of which these terms were reported in the literature. So it helps you to build your own search. You can also look at different abstracts. So once you type in these search terms, these are the types of abstracts that would typically show up. You can look at these different abstracts and then different topics, and it will give you references again on the right-hand side. Once you click on different terms, different keywords again, and some will give you the mesh terms. Once you click on them, mortality, and again, this is supposed to help you set up your own search query, your own Boolean search query. As you might have noticed in the top right corner, there's a little preview. of your what your Boolean search query would look like. So this is a very simple search query. You might want to make it more specific than this, but this tool is to help you start your search query. You can finalize it, and here you can edit it, because this search will import more than 10,000 results. So you may want to make it a little bit more specific, and you can do so here, and it serves as a great baseline to start your search. Okay, so once you have conducted your search, so in this particular nest, they were interested in this particular device, which brought in 284 studies. So after you've done your search, you would then move on to screening. And what's helpful to know is there are different types of screening that you can do in the nest. In order to change the type of screening, you would go to settings. You would scroll down to screening, and you can see that we offer 4 different types of screening. So we have standard, which is just where one user screens each record. You decide whether to include or exclude. We have, you can choose the number of reviewers, whether this is 1 person to review, 2 people to review, and you can play around with what type of screening you'd like to do, depending on what the requirements are for the particular type of project or review that you're conducting. And earlier, Kevin briefly mentioned robot screener. And this is a helpful tool which essentially replaces one human reviewer when you're in a dual screening mode. So this particular nest, we're in dual 2-pass. And what 2-pass means is a screening of abstracts will happen first, and then the screening of full text will happen second. And these decisions have to be adjudicated by a human reviewer. But in order to turn on robot screener, well, when you do turn on robot screener, this replaces a human reviewer at the reviewer level stage. We can definitely go into this another time. It can be a little complicated to go through on a demo when it's not set up in this particular nest. But this is definitely a useful tool when you want to save some time in your screening process. And to briefly show you what screening looks like, whether it's abstract screening or full text, you'll see the abstract, or in this tab here, full text. Oh, this one's not available, but you can read the abstract, decide whether you want to include it, or advance. You would hit advance, which takes it to the next stage, or you can select an exclusion reason here that's already configured, or you can type in another exclusion reason, for example, not relevance already there. Say it's a pediatric study and you're only interested in adult studies. You can add and apply, and it would take you to the next study. So that is screening. So once you have gone through the screening process, this is again where you decide what studies to include, what studies to exclude. Once you've decided what to include, you would move on to tagging. And before you start tagging, you have to create one of these ontologies, a tag hierarchy, as we call it. This is the bulk of your research design. This is where you decide what am I interested in reporting in each of these studies? How do I want to extract that data in each of these studies? And the reason that we have these hierarchies is to make it as flexible as possible. So in this particular mode, we have two modes. We have standard mode for tagging, and we have form-based. And I believe Bajaya asked specifically to talk about a form that you can customize to extract different types of data. This is what we offer in order to do that. So to give you an example as to what I'm talking about, this tag here is devices used, and it has a little Q. That means it's been configured as a question. And then all of these tags that are underneath it serve as a different answer. So for example, We created a tag that was called devices used, and we wanted to ask what devices are used. We selected the question type, which is multiple select, and then created all of these tags underneath for possible answers for this particular question. You can create different types of questions to assign to these tags. So underneath publication information, if you were interested in the objective, you can configure this as a single apply question and add as the question, what is the study objective? You might notice that this particular tag doesn't have any child tags underneath because this is an answer to be answered in a singular text box. Another way you can configure these questions is to have table contents. So once you've created a tag, if you wanted a table associated with it that you wanted to customize, you could go to Contents, toggle on Gather Table Contents, and you can add your own specific columns. And so once you've done this, I'll show you what this looks like when you're actually looking at a specific study. So you'd go into tagging. Skip to one that has a full text. Okay, perfect. You're looking at a full text and you have the questions that were configured on the right-hand side. So the first one we saw was what device was used. We can select the tag, and these are all the options, if you recall, were the child tags underneath this particular tag. So you can choose which is appropriate. Say this one reported the solitaire device. You can apply the tag and it will take you to the next question. Again, to show you what a single apply question looks like, we briefly looked at objective. What is the study objective? This just has a text box instead of specific answers to choose from. We also looked at table contents. Table contents was configured for this tag with schedule, drug type, and administration as the specific columns. You can fill this in as appropriate depending on the study. And this is quite helpful to just have different questions to go through depending on what your project type is. You can customize this and change this at any time. For example, if we were answering this question and one of these tags wasn't appropriate to answer it, you can add in your own tag. So let's say other. Say it wasn't mentioned. We can add this option. It adds another tag underneath. We'd create and we'd apply. So this again allows for extra flexibility. Once you're finished filling out all of these questions, you would hit complete and it would take you to the next study for the same set of questions to be answered. Okay. And so once you've finished answering, configuring this hierarchy, answering your questions, you might want to see what exactly this tagging does, like what outputs are generated and what data you can see from answering these questions. And this you can access in synthesis, specifically qualitative synthesis. So here we have a sunburst diagram of the different tags that were created. So you can also see this in hierarchy form. You might notice that this is exactly the same hierarchy as we saw previously in the tagging hierarchy. And you can select different tags to see which papers reported that particular tag. So for example, if we were interested in what papers reported the headway device as a treatment for stroke, we could click on that tag. On the right-hand side, it would show that this study reported this particular device. You can see all the other tags associated with it and the search that it originated from. Okay, and depending on your project type, you might want to add extra modules. So this is the typical process of a literature review or a systematic literature review. You would have your search, screening, your data extraction, but let's say you're interested in doing a meta-analysis. You might want to go to settings to turn on the module for meta-analytical extraction. So you go down to extraction and you could toggle on data extraction, and the module will appear on the left-hand side here. Similarly, if you were interested in performing critical appraisal or risk of bias, you would go down to the critical appraisal section. You would choose a system. These are the ones that we offer at the moment. But say if you were interested in doing the Sign 50, you would click on that system, and again, it would show up as another module on the left-hand side. And you would conduct these as appropriately. Okay. Does anybody have any questions?
Kindly explain the data extraction part in bit detail.
Dr. Dibjot, can you please be more specific? What exactly you are asking?
Regarding the data extraction part, an example how to do it.
How to configure a data extraction form, right?
Yeah, how does it extract the data in a study, in various studies like which we have finalized the full text.
Great question. Jade, can you present how to extract specific data elements in extraction? I think that we may also want to show the tagging just in case there's different data types people want to pull in. Thank you.
Yep, you're referring to the extraction module?
Yes, I would do this in the I would do this showing both tagging and extraction to show all the options, but I think he was specifically asking about the extraction module.
Yes, okay.
And feel free to jump to one of the nests that has completed extraction to show what it looks like. I think that often helps.
Mm-hmm.
Or Jade and Kevin, if you can, because most of them are doing the prevalence study or proportion data extraction. Because the current nest or the demo nest, they have both RCT or both case control studies, 2R data collection, 2 lakh data extraction form they have. So if you can show just one a prevalence study data extraction, then maybe that will be beneficial for most of them, because they will start with the prevalence meta, then they will go to the higher level.
Interesting. I don't know if we have an open or like shareable prevalence study, but Jade, why don't we show extraction? Do you have access to my stroke demo nest that has extraction already completed?
Yes. Is it this one? Yes.
That one should work.
Okay.
And then let's explain how you extract it for prevalence. I think that the big difference there is that for intervention arms, they would actually need to set up study groups. I can talk through that part if you want.
Okay.
So if you're doing, this is showing a extraction configuration of a study that is extracting a comparison between interventions. The only difference in configuration for that study type compared to prevalence is that when you configure your interventions, you're actually going to just configure the study group that you want to include in your prevalence study. So here you can see it's comparing standard medical therapy to thrombectomy, I believe. Yeah, endovascular therapy to standard medical therapy. If you were doing a prevalence study, instead of configuring a hierarchy that had your comparator interventions, you would instead configure the different patient groups about which you are collecting prevalence data. So, for instance, if you're collecting it about COVID patients who are of different age groups, you would do it different age groups for COVID. If you're doing, let's stay on the COVID topic, if you're doing different comorbidities, you would do a different subgroup under this purple hierarchy for say diabetes, for hypertension, for any other comorbidity that you're collecting separately. Jay, can you show how to actually click on the intervention hierarchy to configure it? So this is very simple, but you do need to plan a little bit ahead. What Jade is doing is she just deleted the interventions that she configured here. And then she's just going to take the hierarchy that contains the concepts that she wants. So in this case, it's the device and drugs that are being compared. But for prevalent studies, it would be the different patient groups. And she just clicks on the highest level tag, and that will configure it automatically. That means that, of course, you need to configure your tags to fit this use case. You need to configure your tags to reflect the study groups you want. At that point, it's very easy to configure these. Just a quick pause there. Does that part make sense? I think we can move to showing extraction. Piyush, is your question about this specific module?
I think PUC is trying since last 1 month.
Actually, I had mailed you also last week about the same. But I just have one doubt, like if in the population, there also we are mentioning the age groups.
Yes.
So how is it different in the intervention group? Like
the intervention group is your choice of the groups for extraction. And Jade, if you go to Study Inspector and open one of the included studies and go to the extraction panel, we can show that. So when you pick your extraction hierarchy, it's going to configure those arms at the top. So you see there it says arms. Whatever part of the tagging hierarchy you identify with those purple nodes will be the options for these arms. So any of the groups that you want to split out. So you might gather age data, right? But that might be across the different arms, or it might be for the whole population. So whatever you configure into that intervention hierarchy, the purple nodes, those are the groups you want. And then the gold nodes are the data elements you want to collect about them. So you should think about going from your tagging hierarchy to your meta-analysis based on the groups you want to collect about, that's purple, and then the data you want to gather, that's gold.
okay, so all of the data will be gathered for those groups only, which I mark as purple.
Correct. Yes, it will only be gathered for the groups that you mark as purple.
Okay, thank you.
Of course, great question. And this can be a bit confusing because we did label this interventions. Nested knowledge was initially used mostly for clinical effectiveness reviews. And so we basically made that assumption in our naming that the groups that you'd want would be interventions. But of course, you can treat them as just study groups. So whichever study groups you want should be identified with the purple, and it will say intervention in the platform. But of course, when you are comparing these, it will simply be your study groups. And then, Jade, do you want to show how to configure one data element? So you can start by clicking in on any of these elements in the hierarchy or in the list on the right. When you do, you'll have the option to configure it as a dichotomous, or just like an event rate, as categorical, which would be like different races. So it'd be like white, Native American, Asian, and so on, or continuous, which is your means and your medians. And for each of them, you're going to have to give some statistical context. So if it's a mean, is it a baseline variable? What are the units, and is it a standard deviation or a range that's reported with it? All of that needs to be configured by you, but that will set it up so when you extract, it will present you with the right information. So for mortality, it will only ask you for the rate of mortality. For age, it will ask you for a mean and a standard deviation if you're collecting the mean, and so on. The only other information you'll need to enter while you're extracting is the time point. If you're doing a prevalent study, I think this is less important, and you might just want to fill in like baseline or 0 days for all of them. But it will just require that you put in a time. You can just keep it as 0 if that's what your study needs. But once you've configured the data elements, of course, you come into this extraction panel. You'll have the PDF on the left, and then you'll have your data that you can collect on the right. And if you've tagged that data, then you'll be able to auto scroll to it by clicking on the little tags next to those. So if you've tagged age, then it will auto scroll you to where it mentions age by just clicking on that little tag that Jade was hovering over just now. So that is our recommended method for extraction. Configure, focus on what are your study groups and what are the data you would need to gather, what types of statistics are they, and then when you're collecting, turn around and simply collect the time point and the data that you've set yourself up to gather from the underlying PDF.
So it is not necessary to have 2 arms, right?
It is not. You can collect a single arm study. The only thing that will change there is that we won't run automated network meta-analysis on top of it, because obviously network in network meta-analysis indicates multiple arms. So the data that you collect will still be completely ready for a report on your subject of interest, but the quantitative synthesis, the NMA chart will not populate. The summary data will, but the automated statistics will not, simply because we need multiple arms in order to run comparisons.
Okay.
Great questions.
Is your nest is ready? That you texted me that you are not able to download your extracted data.
Sir, we were unable to extract. Actually, I was not able to understand how to make this intervention arm. So now I'll modify the tagging hierarchy and then try again.
Okay. Okay, that's good. So Jade, please go ahead. You're next.
Yeah, that was actually all I wanted to go through today. So we can definitely answer some more questions. Or Kevin, if you think there are other things that we should go through.
Would love to throw to questions first, just so that we know what the pain points are or what specifics that your team is concerned about. There are a couple other features that we could show. I would love to get a little more into Study Inspector. Jade is the only one that I was thinking of. So first, open it up. Yeah, Dr. Lamba.
Yeah, I just want to know how the extracted data would look like. It would convert into an Excel, or like, for example, the age was for a single paper. So they will combine all the papers that have been finalized to get a mean value, and then the extraction would be in an Excel file or what?
Great question. Jade, can you just go back to that same nest and show both quantitative synthesis and how to export? Maybe even, and this might take a little bit longer, but I think showing Study Inspector will help with that, right? So we can show what Study Inspector does, including download.
Yes, okay. So Study Inspector is a module on the left-hand side here. And this shows a list of all of your studies in the nest. You can use these filters to filter to different groups of studies. So if you only wanted to see the studies that were included, we know that's only 4 studies, we filter to included. And so we wanted to download all of the extracted data for these 4 studies. We would go to the right-hand side, hit download, and we have these different options in the drop-down here. We can download a CSV, a spreadsheet of all of the studies, a RIS file or a full text PDF. You can download screening decisions, the tags that were applied, the extracted data, and so on. So in this particular case, everything that was performed in the extraction module would be downloaded when you hit extracted data. And you hit download, it would download the CSV file. And I'll open it up just to show you what it looks like. And so this shows you all of the quantitative data extracted for all of these studies, depending on what was tagged and what was extracted. So that is a spreadsheet of all of the extracted data, but in terms of the quantitative output, that's in our synthesis module, you would go to quantitative synthesis. And as Kevin mentioned, this is where our network meta-analyses outputs are generated. So previously we saw the qualitative outputs, that was the sunburst, and these are the quantitative outputs. So here first we have a summary, our 2 interventions on the left-hand side. You can open these up, see the specific studies, compare different outcomes, different baseline characteristics that were tagged. So if we're interested in mortality, we can see the populations, the total percentages, the confidence intervals, and we can compare them to different data elements. We can also look at the distribution. So if we're interested in how 2 specific data elements compared to one another, how they were reported in the studies, you can change the different axes. So again, if we're interested in mortality and how it compares to males and mortality, each of these points represent a different study. and intervention arm. And then we have the network meta-analyses. We have the odds ratios. If we want to open this for, again, mortality, we have the odds ratio. We have different forest plots. And again, how different studies fared depending on the intervention arm. Okay, so they are the main two outputs that we have for data extraction. Okay, do we have another question? I think I saw another hand up. Yes, Dr. Lambert.
Yeah, so from what I've understood, tagging is the most important part for data extraction. So can it be explained a bit in detail regarding the tagging part for various like for the Pico?
So
An example.
how would you tag for, if you know your Pico, how would you set up tagging and how would you tag the underlying articles? Jade, I think you can take that, right?
Yeah. Okay, so this is a pretty straightforward hierarchy, and I think it's important to point out in the previous nest that I was looking at, we were in form-based mode, but that's optional. You can also stay in the standard mode, which is the way that this hierarchy is created, is configured. So we would create a tag for each of the Pico, each of the letters in the Pico. So we have population, we have interventions, and we have the outcomes and comparisons. These would serve as your what we call parent tags. Underneath each of these tags is where you get a little bit more specific into what you want to report. So we have population, and underneath population, we're interested in the timing, the medication, and the demographics of each population. So, and underneath demographics, we have age, we have the sex.
During the extraction, we'll have to, these are just, tagging is just a way to like highlight whatever is important in the paper. And during extraction, we have to manually enter that in the.
Yes. Yeah, exactly. So when you're tagging, you highlight where it is in the paper, and then in the extraction module, that is where you would start to put in the numerical data. And the tags are helpful because when you're extracting, you can click on this icon here to see what was reported in the text. So you can see here.
Also, really quickly, I wanted to fill in. Tagging does not just set you up for extraction. It does help set you up for extraction because it does enable that auto scroll that Jade is demoing, but it also builds your qualitative synthesis and it's exportable as Excel. So if you want an Excel document containing, or an Excel sheet containing all of the text from underlying studies organized by tags, so basically columns are tags, rows are studies, and then each cell will contain the exact text that you enter or extract. So it's not just to set up its extraction, it's also to effectively get your systematic review data that it's qualitative. If it's not meta-analyzable data, then you still might want to collect it. You still might want it in an Excel sheet. So it's multi-purpose.
So data would not come by itself. We have to add, like for age also, for the individual paper, we have to
Yes.
add.
Yep, correct.
Okay.
The actual collection of the data, we are working on that. So I hear this question a lot. We are working on effectively trying to auto-populate the data for you, but
Yeah.
we have to have absurdly high accuracy standards before we roll that out into a product. So
Yeah.
as of right now, extraction is manual with the assistance of that auto scroll. And I now see 2 more hands up. Devashmita, do you want to jump in?
Am I audible? Hello.
Hello.
Am I audible?
Yep, we can hear you.
Sir, so what I'm understanding that this tagging can help me to create a spreadsheet or to create an Excel sheet, which is the master file of my systematic review.
Yes.
Can we interpret it like that way? But sir, in kind of a simple prevalence studies for which we can do a master table for our systematic review. So, say for an example, in a variable like age, some paper give some article give represent the variable age in a percentage, not in a count, or some paper give it in a mean. So sir, how can I standardize the variable age through this tagging?
Great question. So you can do it one of 2 ways at the tagging stage. You can either create a single tag for age and then simply collect every way that they actually report age under that as a tag. So not using the extraction module, but instead just collecting age as whatever text is from the underlying study. And that will be exportable. So if you capture in that text that they report, like the mean age is 69 and the standard deviation is 10, that would be captured in the text of the tag. You can also make subtags. So you could have age, and then you could make age mean a subtag. You could make age bifurcated or dichotomized. You can make age median a subtag, and then you could tag under those. The advantage there is you're breaking it out by the type of data that's reported. So one way you get it all simple under one tag, and you capture the context, like what type of statistic is this in that little text excerpt that you extract. Or you create a subtag for each way that the data is reported, and that just depends on your preference. Are you looking to summarize it all in one column really quickly, or are you trying to break it out by the type of data that's reported and do approach number 1 if the former and approach number 2 if the latter? Does that make sense?
Yes, sir. Thank you.
Great question. And I do want to add a piece there. Your systematic review should be structured based on the tags that you put into the hierarchy. These tags will be the columns in your Excel table. They will be the tags you can turn into data elements, and they will also be what informs your synthesis diagram. So when you're creating your tagging hierarchy, think about what you want your columns to be. Think about what you want your master table for your systematic review to look like, because you can export your tags to create it, But if you didn't set up your tags to populate the data you wanted, it's going to be extra work to make that table. Aravind, you have a question?
Yeah, thank you, sir. If I'm audible. So this
Oh,
question is,
you're quite faint, Jade. Are you able to hear him?
No.
yeah, am I audible now?
Yes,
Oh,
Yes.
much
much-
better.
Okay, sorry. Thank you. So my question is regarding the format. I mean, since tagging seems to be a very important step in terms of determining the characteristics as well as data extraction. So when do you recommend to tag the studies, whether after the full text screening or during the full text screening itself?
Wonderful question, and I can tell you understand the system by asking it. You can tag during any screening. So the tagging module, Jade, if you jump to screening, you will always have the option to add tags at that point. However, I would only do so if you already understand that content very well and know exactly what you want to tag. My preference is to wait until after full text screening, because then I know exactly which studies I need to tag. Excuse me, I needed to sneeze briefly. So I often will only do it at the tagging stage. So I will do my abstract screening, I'll do my full text screening, and only after that will I tag. But I always have the opportunity to add those tags earlier. It's under the screening panel. So if I wanted to, I could include the study, but before I do, I could add a tag for study type. I could add a tag for the population of interest and so on. And then Vijay, I saw your hand up as well.
Yeah, I think this question partly answered my queries. So just a clarification, do I have the liberty to change the tag? For example, I have a primary outcome, but while doing the screening, I find another outcome which is interesting and I thought I should record it. So do we have that
Wonderful.
option?
Yes,
While-
so this is true for both screening and for tagging. You can configure them on the fly. So Jade, why don't you make a tag for something we don't have yet? From a study. Yeah, so jumping in, she's in the tagging interface and she's just going to start typing a new tag name. Yeah, like background is not a tag in there yet, I think. So you could just start typing background. And then you see it says add option. That will enable Jade to not only configure that tag, but also pick where in the hierarchy it should live. So if that's a study characteristic, she could put it there. If it's a population related element, she could put it there. And then that will add that to the hierarchy on the fly. So you can basically change the contents of the hierarchy as you go. So she's now added the tag background to the hierarchy and she can collect it. Similarly, when she's screening, she can actually do the same thing. She can add a new exclusion reason while she's screening without needing to go to the configuration page.
That's
And
wonderful.
you do that the same way. You just start typing it. So you can see here she's typed in new exclusion reason, and it will offer her the option to add and apply it.
Great features.
Yeah, that one is so helpful. You can really, as long as you have a picture of the tagging hierarchy in your head, you can really just work on the fly. Okay, Gatish?
Yeah, I have a similar question. Like when I search through this search exploration, after hitting the search button, if I go to tag section, I'm already seeing some of the auto-generated tag. So if the system is also generating auto-generated tag, or it's just some dummy tag that we can see and edit.
Interesting. So when you complete search exploration, if there are tags there, are you sure they weren't created by your team? Usually we do not build out the tags without you seeing them. So it might have been from your team, but those are editable. So you can jump in, and if they're not exactly what you're looking for, you can simply change them. Yeah, you're asking basically, was your tagging hierarchy built out from your search exploration? Right?
Yeah, it's based on the search exploration only, or it had been manually like prepared.
It was likely manually added. Perhaps someone imported a template hierarchy because it's very easy to just grab existing hierarchies and import them. So it might have been that someone was using a template, but I doubt that was made by your search exploration. That should have been only based on your tag building. But any of those tags, you can edit them by just clicking in on the tag and changing its contents or changing its parent.
Okay, thank you.
Great question. I think we have time for a couple more. We have 5 minutes left in this session, so please feel free to bring up any further questions.
So friends, do you have any questions? And I believe that once you use this, you will have a lot of questions. This is a very excellent software, and in one screening or one study, you will find you can extract many data. Most importantly, the qualitative information that usually we miss for our meta-analysis. We focus the quantitative, but most of the time we are not able to capture the qualitative informations. So this nested knowledge will help on that aspects. And the diagram it generates, the sunburst diagram, it's a very amazing diagram. So both Kevin and Jade, thank you very much for your time. And let us use these features maybe 1 week or 2 weeks. And I hope there will be many questions and we'll get back to you. And if you have time, just if you can show the new features it is added there, the HK component, just a quick glance.
Yes, absolutely. So first of all, actually, thank you guys for a wonderful session as well. It always comes down to good questions and good listening, and I could tell that you guys were. Jade, if you just want to present quickly the HTA demo nest that you've built. So there aren't new features that are only for HTA nests. Instead, we've built out template hierarchies that capture HTA questions. So you can see in Jade's hierarchy here that she actually has put together the critical appraisals that are needed and the evidence that is required from, I think this was built on the NICE standards. So this is a nested knowledge built hierarchy that reflects the HTA requirements of NICE in the UK. And so if you want to use this hierarchy, talk to Jade and she can get you HTA templates for that sort of collection. And those can be any of the types of HTA review, which I think are usually the burden of disease, clinical effectiveness, and then economic. We have templates for each of those. They still use the same nest structure. So you don't need to change your nest structure to do these. Instead, we simply try to set you up with the right tagging hierarchy to get underway. So it's a template hierarchy that we can share with you at your request.
So basically, this feature is inbuilt within the existing nest, whatever we have access to.
Sorry, say again?
No, this HTA feature is actually inbuilt within the systematic review meta-analysis nest itself.
Correct. Yeah, it's already within all of the nests that you guys can create, but you have to add it as an imported hierarchy under the import hierarchy right there. You may not have access to it right now because you need to be a member of an organization that has access to it. You're seeing actually in this case the nested knowledge templates. These are those that we've created for our uses or that we share with clients. So your organization will have to build out your own, but we can also share them with you. So when you've got your organization established and everyone has joined it, you can reach out and Jade can share these hierarchies so that your organization can use them as well. Your organization can also build them, right? So if you have a different type of review that you want to build a template hierarchy for, you can also build that for your entire group.
Thank you.
Wonderful. Any other questions that we should cover? Oh, I saw one question in the chat here. Jade, can you quickly show the manuscript editor? Maybe like a one minute demo of the manuscript editor.
Oh, this one doesn't have a manuscript. Okay, let me go into this one. Okay. Is my microphone on? Yes. So in the manuscript editor, this is essentially the same as any Word editor, Word document, but what it does allow you to do is add in custom tables. So see here is a custom table based on the studies and tags in your nest. What this looks like, in order to customize it, you would configure these different drop downs, add specific columns. So if you were interested in a column for DOI, you would add that column in and it would update and add a DOI column. So that's quite helpful to have as your custom tables. All of this is, of course, exportable. If you go to the top right, you can download a Word document, which you can, again, take out of the software. So none of your data is ever trapped in the software. As well as a custom table, you can also add in a Prisma diagram. Oh, yep, here it is, that was generated for your particular review.
And then there's one more question, but really briefly, you can also manage citations in your review. It's not as built out as EndNote or other citation managers. So if you are very concerned about citation format, I recommend exporting this as a Word document and citing it using EndNote. But we do allow you to, in one click, add any citations from your nest to that Word document. And then there's one last question that I'll just pick off from Piyush again. I do not actually think that we have a built-in tool for critical appraisal of prevalence studies. Now, there are several options. If you find one that you want us to build in, please send it my way. You can just email it to Jade or to me, and we will look into adding it. And then if not, you can always build those questions into a tagging hierarchy. So Piyush, if you have a critical appraisal for prevalence studies that has only a couple of questions, you can always build those out as questions in tagging. And yet again, that's something you might want to reach out for help your first time you're doing it, but you always have that option to effectively customize the questions in your tagging hierarchy to any critical appraisal of interest. And then just going to pick these off quickly because we're a little bit over time. You can consider that tagging is a lot like coding the qualitative data from a study, but also recognize that it has flexibility to what you're actually pulling out. So it could be an exact code for a given disease, or it could be an entire sentence from the underlying study. What it does is it attaches both that excerpt and the label to that underlying study. So it's always going to be filterable, it's always going to be exportable, and usable in that sunburst diagram from that. So it's a little bit like coding, but it has the addition of that excerpt that that's attached to the, so it's a label that's attached to the study, but it also has the evidence in that excerpt. And yeah, thank you, Vijay. We are so happy with your team's use of the tagging and qualitative feature. Thanks to everyone for your attention. Jade, is there any last comment before we sign off?
No, just thank you. Thank you everyone for joining, and we'll definitely get a link out to Kevin's demo, the webinar link for everyone to watch for a more detailed step-by-step on how to use the software.
All right.
Thank you, Kade and Kevin.
Absolutely. Thank you.
Okay, thank you everyone.
Talk to you soon.
Bye.
So those who want any doubts, you can stay back and ask me questions. If anybody has any questions regarding our conference or how to start your work, you can ask me.
Good evening, sir.
Yes, please.
Good evening, sir. This is Dr. Pallavi. Sir, I want to start the work. I have not started yet. So, how to begin, sir?
Do you have a research question?
Sir, not yet, but I can come up with one in a week time.
Okay. So first, as I discussed in the previous session,
Yes, sir.
you have to identify research question, and that research question, because we need data for our meta-analysis, either
Yes,
qualitative or quantitative. So what I said, you have to look for a quality journal. Okay. Either Lancet, JAMA, BMJ, those type of journals, but don't go beyond that, like Frontiers, MDPI, PLOS, don't look those journals. Okay. So once, now the second step is identify minimum four articles where you want your data to be extracted.
Yes, sir.
Either it is quantitative or qualitative. Once these three steps is done, first identify question, look for journal articles, find 4 journal articles that published recently, maybe 3 to 6 months of time. Then once you complete this, then you contact with our team. Sir, I have completed these 3 steps. So how to proceed?
Yes.
Okay. So these are the requirements, because we have ample time. Our systematic review meta-analysis will start, the session will start in the month of, I think, October. But today is, it is June. Maybe most of the people, most or say 1/3 of the people, they will start their meta in the first week of July. Okay, we have, because I am also not familiar with this nested knowledge because it is a very new software. For us, it is as like you, it is same for me also. First, I have to learn its all features. Then only I will be able to demonstrate you. So today they have just shown very briefly. They posted the YouTube videos for each and every aspect, like how to screen, how to search, how to extract. So every component they have posted there.
Yes.
Okay. So basically, I came across this nested knowledge just accidental as a coincidence, you can say. So we tried with Covidence, AP review, CIS review, and so many softwares. And once I was going through all the YouTube videos, then I find it is an excellent software where you can extract all your quantitative and qualitative data. Because once you are reading a manuscript, there are many information there.
Yes.
Maybe you are aware or not aware. When we started this meta-analysis journey last year, okay, we got one article from NEJ, New England Journal of Medicine. Okay, from that one journal, we generated at least 4 or 5 research questions, and we published all those 4 or 5 research questions in a full fledged meta-analysis. So, what my question is: once you are reading one article, if any primary outcome, secondary outcome, or anything you come across, just tag it. Okay, extract all those data. So later on, you don't know when the data will be useful. So in one labor, one time, so you are extracting many information. Then use accordingly.
Yes, sir.
So that is the good advantage in this software.
Thank you, sir.
So what are the important steps after data extraction?
It
How
is
do we
same
have to
like either you can use their inbuilt system to generate the forest plot, the price chart, or you take out the Excel sheet and do it in any software like Stata or SPSS. It's up to you.
and for manuscript, they give some information like introduction and then discussion, or that we have to build on our own
No, everything you have
from
to
the
build
study.
your own. Everything built, you have to build your own, but they are just making your life easier. For example, if you want to insert a price chart, you don't go back to a software and build your price map, right? So you just click the price map, it will be inserted in your manuscript. Okay. So you
Okay.
want basic characteristics of a table in your result section. You click on the table, what are the features you want to add, select those features. A table will be placed in your result section.
So statistics they will add or what?
No, those
Statistical
type of
para.
statistics. You see, in meta-analysis, what statistics? Meta-analysis, it is the pooled synthesis or pooled evidence. Okay, the statistics they will generate, but if you want a higher quality graphs like the R or Stata produce, so you see their graph is not so good. Okay, so in
Okay.
that case, you extract the data in Excel format and use your software, or later on, if you can ask them to integrate the R code. Okay, so that can be also done.
Okay.
So let us start one by one. Okay, first start with the search, tagging, extraction. So then we'll come step by step.
Hello, good evening, sir.
Yes, please.
Sir, I'm Dr. Dhruv Kapoor. I wanted to ask you, sir, regarding a search of a research question, uh, the 4 articles that we pointed out that we should collect for quality data, 4 articles which have recently been published. So, what do you suggest that all 4 of them should be in one of these reputed journals like Lancet and JAMA, or one of them in that and the others recently published but in some other journals?
It is very good if you will get 4 articles from those journals. Then your meta paper should be published in those type of journals. But even if you are not able to get 4, at least if you will get 1, then that will also suffice.
Okay, sir. Thank you.
Okay,
So there's one
you
question.
don't worry regarding the research. Those are new. Those are old. They can generate their own research question and article. But those are newcomers. Don't worry. For the fast meta paper, our team will provide you a research question and that 4 articles. Those are newcomers, you should not be worried. Once you learn how to do it, then the second paper you will do it on your own. Okay, Dr. Arvind, you supposed to say anything?
No, sir. I was just pointing towards the chat box. So there were a couple of questions. So one is from Dr. Himanshu. So he's asking whether we can collaborate on dental related topics. Yes, any health related topics we can collaborate. So that should not be an issue.
So any question, friends? So my suggestion is, please go through the YouTube video posted by the Nested Knowledge. Just go through it. Be familiar. Take 1 week, 2 week, 3 week. You have ample time. 7 months of time you have. Okay, do not worry. You should learn all these features. It is an excellent software. Means why I say it is excellent, because it will reduce your workload. Okay, one paper you are reading, you extract as many as information you like. You don't know whenever it will be useful. Maybe your plan is one research question, but while you read that article, why I'm saying you look at those quality journals, NEJM, Lancet, because you will find many information in those articles.
Good evening, sir.
Yes, please.
Sir, my question is using nested knowledge literature search, we don't need to search in all the major databases separately, or?
No. You see, Nested Knowledge has a API link with the PubMed, because PubMed do not charge anything. Other
Okay.
databases, they are charging very heavily, so Nested Knowledge cannot do automatically. For
Sir,
that, you need to do
search
a
for
search
PubMed on,
with your.
and we need to conduct separately for other databases.
You need to conduct separately and upload those data.
Okay, sir. Okay, sir.
Or like PGI have a data. If PGI will give the API, then Nested Knowledge will be happy to integrate that API. But it is very difficult, because PGI, I don't think they will give the API to students or us. So it's better to do a manual search in our database and upload it.
Okay, sir.
Okay. But if you are doing a living systematic review meta-analysis, it is always better to use the Nested Knowledge PubMed system, because it is an excellent feature. That is only available in Nested Knowledge, not any other softwares like others available in the market.
Yes, sir.
But we are not planning any living in next 1 or 2 months. First, let us start with a prevalence study, then say a comparison or an interventional study, then a network meta-analysis. So these are step by step. Those who are newcomer or beginner, you cannot start a network meta-analysis on day one. You start with a simple meta, single arm meta. So see how the data is extracted, how the forest plot is generated. Then only you go step by step.
So what is a living review?
Sorry, Dr. Debjot.
What is a living review, sir?
Living meta-analysis. Living meta-analysis means you designed your search algorithm. Okay, you posted there, and for example, after 1 month, one new study added on. So you don't need to do again search. So that study will automatically add to your system. Okay, and it will reflect automatically to your Prisma chart. And if your journal is allowing to update your data, so the result will be also automatically updated. Like during the COVID time, the knowledge is gradually evolved. So at that time, BMA as a living meta-analysis, I was following that meta-analysis. So all the outcomes, it is automatically, because they have a API tie-up with a software. So it is the forest chart, everything means changed with the progress of time.
Sir, I think even in living meta-analysis, manually we have to verify the study and add the extraction. Only after that it gets updated, I suppose.
Yes, that is one aspect. Okay, friends. So I will post you the next meeting schedule. Let us first take 1 week time. Be familiar with the nested knowledge features. This 1 week till 1st of July. All of you, my request is whenever you are getting time, just familiar with the different features of the nested knowledge. Because next 1 year we are going to use this software heavily. Okay, so good night. If anybody has any specific question, ask me. Otherwise, we'll stop this meeting.
Hello, sir.
Yes, please.
Sir, I'm Sumit from BHU. I have a question that is for meta-analysis, is data will suffice or I need to learn R also?
Okay. So if you are learning the meta-analysis, this nested knowledge is enough. You don't need any other software. So this is the one advantage of nested knowledge. You don't need R, spaces, or stata. But if you need a high quality output graphs or figures, okay, we are teaching R, because the first workshop will be how to write your manuscript, the July 7 to 9. The end week of July, we are teaching the R, means all those software packages required for meta-analysis in R. So don't worry, this conference is a complete package. From starting how to start with your manuscript writing to all advanced features of meta-analysis using R.
Right, sir. Thank you.
Why we are teaching R? Because R is open source and no, you don't need to pay anything. Whereas in Stata or Spaces, you have to pay a heavy amount.
Right, sir. Yes. Thanks, sir.
So any question, friends? So thank you. So let us close this meeting. I will share the recording. I will share the recording shortly.
Thank you, sir.